and I'm going to introduce our speaker for the day. Today we have Abhishek Dutta, Assistant Professor in the Department of Electrical, Computer, and Biomedical Engineering at the University of Pittsburgh. Abhishek received his MS from the University of Edinburgh and his PhD from Ghent University, followed by a postdoc at University of Illinois. And since 2016, he's been assistant professor at University of Connecticut, where he was got an interesting award, the recipient of the AI 2000 Most Influential Scholar in the Field of Multimedia and the Informatics Prize for Outstanding Thesis from Edinburgh in 2007. Dr. Dutta uses control theory, deep learning, and optimization techniques to understand and regulate infectious diseases and complex disorders. So with that, I'm going to hand it off. Abhishek, the floor is yours. Please take us away.
Thanks so much, Harry, for this wonderful opportunity, and a very good afternoon to all of you in whatever time zones you are. I'm at Eastern. Yeah, that is me. So my name is Abhishek, and apart from being a professor with electrical, computer, and biomedical, I'm also a professor at the Pratt & Whitney Institute for Advanced Systems Engineering at UConn, and I direct the Dutta Lab. Now I'm gonna first briefly introduce sort of the main themes in my group that we look at. So the first theme that you see to the right top is biological and robotics. So this biological robotics is based on controlling the brain. I'm going to briefly touch upon that, on how can we control at least some functionality in brain, and how can we actually realize it on at the level of insects, if not at the human scale. So this is the biological, neurological, robotics track. Now the second track in my group is one of the central themes that we are, all of us alongside my PhD students, we are all looking at. This is the fascinating field of network medicine. So in network medicine, we look at diseases as a network interaction at various levels, like in principle at the biomolecular level. So how do biomolecules at the level of genes or proteins or transcripts interact with each other? And how do diseases change this dynamic? And how can drug actually rectify this dynamic and bring it back to its original healthy state? So in this track, this is the ocean of knowledge and sort of opportunities. I mean, the human network of diseases and human network of biomolecules is so vast. So one has to look at it more objectively in terms of one particular disease or interaction of few particular diseases. What is the mechanism of action of drugs given a certain disease, and so on and so forth. Again, I will briefly touch upon that. However, the central theme, as we understand today, is in fact this systems epidemiology. And I have been a proponent of introducing this term to our community, which is systems epidemiology. Although this term existed in the past, but after the pandemic happened, I was very strongly advocating for this term at multiple levels, from at NSF to NIH to IEEE to Nature, at various levels wherever I could. Now, what do I mean by systems epidemiology? Well, by systems epidemiology, I mean that we need to understand the proliferation of infectious disease at two scales. So one is at the larger scale, which is at the society level. So where I want to see how the infections actually are transmitted in the population, and how can I control this by a systematic feedback control. So now the control variables in the society itself are the interventions, right? At the interventions at the public health level. So this is one level. And the second level that immediately follows is inside our body, which is also called in our community in-host. So the diagram that you see here shows that given any new infectious disease, which happens to be, in our case, the coronavirus. So how can we understand its dynamic relationship with our immune system first? And second, how can we then use some of the existing drugs? How can we then repurpose in a more systematic way? You see, repurposing to me doesn't mean that, you take a drug from the past and you just see how if it's efficacious or not. No. That's not what I mean at all. So what I mean is you find out what is the exact dosage and scheduling of that drug, of that antiviral for the new disease, which is COVID-19 for us. So this is exactly what is going on in that third figure. And integrating the in-host control system, in-host meaning I want a controlled therapy, a controlled antiviral therapy. This is what I mean by in-host systems epidemiology. And at the public health level, what I mean by the systems epidemiology is how do you find controlled interventions at the scale of population. So how I want to define systems epidemiology is by the integration of these two mechanisms. Okay, and so roughly my talk will go on in this particular order. So first we will look at the brain control problem. Second, we will look at the network medicine problem. However, I want to get those done in around 20 minutes because the central theme of today's talk is again, systems epidemiology, which I want to spend a little bit more time on that. And kind of at the 45 minutes mark, I want to end and open up for discussions. So here, so what we have developed alongside some of my PhD students who are there today is I took a roach, that is the platform vehicle for us, which is the insect brain. So then what you see here is we want to perturb the brain dynamics of this insect using two kinds of stimulation. So one, we are stimulating the left lobe of the insect brain, and the second, we are stimulating the right lobe of the insect brain. Now, what we need to figure out is what kind of stimulations or what kind of frequency should we inject into the brain of this insect in order for it to obey certain neuromuscular commands from us. For example, I want it to turn left, or I want it to turn right. I want it to stop, or I want it to look for food, and so on and so forth at that level. So you can see in the video here that this is so my students work in the group. So the insect has been asked to sort of make a left turn, and after certain resistance, it is actually going to turn to the left, and it will. You see, it stops, but again, it continues the turn to the left. So how are we achieving this? So we are achieving this by stimulating the right part of the brain of the insect. And it kind of makes it feel that there is an obstacle to the right, and therefore I must turn to the left. So this is one of the very, the mechanism that is exploited at a very gross level. However, much more can be done on top of that. And I want to show that a little bit more by going a little bit deeper into what is happening inside the brain. So our brain, or an insect brain, is composed of a vast network of neurons which are connected with synapses. Now, what happens is everything in the brain works by something called as a spike, meaning a neuron spikes, that is the voltage of a neuron spikes, which is exactly what is visible on the graph here. So what happens is there is a resting potential in the neuron, which is at around minus 75 millivolts, and then it spikes, so it polarizes to positive potential, and that it immediately again comes back to the negative resting potential. So this is defined as a spike. And neurons talk to each other, communicate with each other by spikes. We really do not know any other thing I mean, we do not know. I don't want to say that this is the end of the story, but till now in computational neuroscience is based on this spiking behavior. So when various neurons spike, what happens is they develop strength in the synapse. By strength, I mean they develop new connections, or the connections become stronger and stronger when these neurons spike. And by this, formation of memories takes place. Not only formation of memory, but reasoning, decision-making, everything that we do. Right now, computational neuroscience tells us is all that happens by when the neurons spike, okay? So then the central question is when the neuron spikes. So this can be explained by a simple, not a simple, it's a nonlinear dynamical model. What happens is the neuron collects information coming in from dendrites. That is the information that is coming in from other neurons in the brain through the current injection, okay? Now, if there is sufficient current that is being injected into this neuron, what it leads to is it increases on the left-hand side, if you see the membrane potential, it leads to an increase in the membrane potential. So it increases from its resting potential, which is around minus 75 millivolts, to minus 55 millivolts, which is called the threshold potential. So from minus 70 to minus 55, once it increases, you can see it clearly, then a spike must happen, okay? So this happens by an exchange of sodium and potassium through the membrane of the neuron. Okay, so then our objective is to make the spike happen. And clearly what it means for us is then that we should make this jump from minus 70 to minus 55 millivolts happen at will, right? How do we do that? So then obviously we need to manipulate this current injection here. So then the question is, how should we manipulate the injection of current in order for that spike to happen? And the spike happening and not happening can be linked to multiple neurological disorders. Okay. So I want to quickly introduce this thing, which is one of the central themes of our group and my own research, which is called model predictive control. Now, what model predictive control means is that we have a model, which in my case always is kind of a dynamical system, right? So dynamical system model looks like x of t plus 1, where x is the state, is a function of the previous state x of t and the control input u of t. So in the case of the brain, the previous state will be its previous membrane potential, and the input is going to be the injected current. And that, because of that, it evolves into the current state, which is the current membrane potential, and so on and so forth, right? This is a typical dynamical system. So if I have this model, which I wrote down here, right? So this is the model of the neuron. So if I have this model of the neuron, what I want to say is then I can predict its response in the future, correct? Because if I change the current injection, I can predict what the membrane potential will look like in the future. So this is the model prediction part of this model predictive control. Okay. Now the second part is, of course, I want the change in the trajectory of the membrane potential to my liking, right? And what do I want? Well, I want it to go to the threshold potential. So which is my reference target? Okay. Then I take the difference between the target which I want it to go to and where it actually would go to. So that becomes the error. Now that error, based on that error, I design an optimization objective function whose objective clearly is to reduce the error, right? So based on that optimization, it splits out the desired control actions which must be taken in the future in order to decrease the error. And so those are the control actions which will be what will be my future current inputs that I must inject into the neuron so that the neuron actually spikes, right? And this loop continues. So this is exactly what happens in a model predictive control scenario. And this is very important for us in terms of manipulating any drug. Here, you know, what is the drug here? The drug here is the current, right? The drug can be oxygen, like we know in emergency. The drug can be actually a drug infusion, right? So what happens if I want to manipulate, for example, the drug infusion for us, let's say remdesivir, right? I want to manipulate remdesivir. So obviously I must have the model then changes from a neuron model to the dynamic of the virus with respect to the drug. So the drug is the control input, which is the antiviral therapy. And the state then is the evolution of the virus and the evolution of the immune response. Okay. And what will be the reference trajectory? Well, obviously, I would want the virus to go to 0. So that will be my target. And then based on that, I'll form an optimization function, whose job will be to reduce the error, meaning whose job will be to reduce the virus. But not only that, its job will also be to reduce the drug injection. Okay. So the best part of model predictive control is that it can take care of constraints, meaning for the brain stimulation, I cannot inject as much current as I want because that is going to fry the brain. So therefore, there must be a constraint on the current. Similarly, I should not be able to inject as much antiviral therapy because that leads to toxicity. So therefore, I can, constrain the amount of drug that I can use. So the optimizer then optimizes that what will be the best sequence of drugs that should be injected to the body in order to get a certain amount of viral deceleration. Okay. So this, what you see here, is the drug infusion in the future. And then you apply those step by step. And then again, you see, okay, well, is the objective achieved or not? So this is the overall model predictive control, and I'll keep coming back to it. So therefore, it is very important for us to understand what is model predictive control. Okay. Now, if I apply the same, which is work done by my PhD student, so Rom Ting Yu. So if this, you apply this logic to this problem of neuron control. So you can see this here. What happens is I have this Hodgkin-Huxley model of the neuron, which I just explained, right? Now the input is the current and the output is the membrane potential. Then you take the error, you compare with the reference. What is the reference? The threshold where we want to go. And based on that, you compute the controller, which is in this case is the model predictive controller, computes what should be the ideal current injection. Okay, but note that there is a noise. What is the noise? Well, there are lots of neurons in the brain that are firing at random. So that is the noise. So in spite of the noise, my control must be robust enough to cause this spike. And you see the results here. And you see these spikes which happen in spite of the noise in the brain. And the red line shows the current injection, the controlled current injection that takes place, which actually causes this spike to happen. And you see the membrane potential rising from -70 to -55 millivolts. And then the spikes actually happen. So this is the application of an advanced control technology to control the brain, in this case, a neuron. Okay, so this is the end of the first part of my talk. And we'll now quickly go to the second part, which is the part of network medicine. Now here, the first thing I want to introduce is learning mechanism of action for new drugs. How do you do that? Okay, so Harvard and MIT people have done an extensive experimental study, and they have provided the data. Of course, the data is obfuscated. Okay, we literally don't know exact things about the data, but still the data exists. So what they did is they took like around 20,000 drugs, and for each drug, they took 100 cells and 772 genes. Okay, so they measured the gene expression of 772 particular genes and the viability of 100 cells. Okay, in culture, of course, in vitro. So they applied these drugs, 20,000 drugs, and they did it 20,000 times for each of those things. Okay, now there were particular dose, type, and duration given to that. And these 20,000 drugs, we knew what were the mechanism of action for these 20,000 drugs. So from this input, we knew what the drug mechanics were. So for example, the drug could be an antagonist. The drug could be an agonist, right? And so on and so forth. So we knew. So these were like 200 mechanisms of action that were known. So what I did is I took this data And I fitted a deep learning model, a pretty sophisticated deep learning model, which had many dense neurons and a few layers. And it had leaky ReLU and batch normalization and dropout regularization. And finally, you can appreciate that this is a multi-class classification problem, because given this signature of gene expressions and viabilities, what I am predicting is then what would be the mechanism of action of the drug. Okay, so then I learned this on these 20,000 data points that were provided to us, but obfuscated. What do you mean by obfuscation of the data? Is that they did not reveal what drug that was, right, for their own reasons. So, however, I trained this, and what it means is that now if we have a new drug, and then you take the same 100 cells, and you measure the same 772 gene expressions over the new drug, I should be able to predict what is the mechanism of action of the drug. Okay. So how this works is, of course, this is how a deep learning works. So you have the input gene and cell data, which is passed through this network of weights, and that predicts the drug mechanisms. This is what the forward propagation is. And backward propagation means that you have a cost function here. That is, I want to match the drug mechanics with the true drug mechanics, right? So I want to minimize the difference of my predicted drug mechanics with the true drug mechanics. So that is my cost. I want to minimize that. In order to minimize that, what should be the best sequence of weights, right? So that we determine by pushing these gradients back. So this is what the this animation is showing, that I'm pushing these gradients back. So if I do that, you can see next in the plot that by doing that, I can actually minimize this loss function, which is the difference between the predicted mechanics and the true drug mechanics. And my accuracy increases. Okay, so this paper is published in IEEE MBC, those who want to check out the details of it. But this is how new drug mechanism of actions can be predicted. Now, the other part of this is very fascinating. Now, here what I want to introduce is a concept called graph neural networks. So the idea is that I want to generate new drugs, right? I want to generate new drug molecules by training a graph. So I want to consider a drug molecule as a graph, first of all, okay? So what do you mean by that? So a molecular graph is composed of the atoms. Now the atoms are denoted by X, and an adjacency tensor, which is A. What is the meaning of adjacency tensor? It means that which atom is connected to which other atom. That is all it is, okay? So in this way, I can describe any drug as a graph. So where the atoms are the nodes of the graph, and then the adjacency tensor. This is how the atoms are interconnected in the molecule of the drug. Okay, so you can see this animation here. What it shows is then you see these orange bars. So these orange bars are the feature vectors of the atoms, right? Which is X. So what can be the feature vector of the atoms? Well, it can be its molecular weight. It can be if it's aromatic compound or not. It can be its charge, polarity, and so on and so forth. So each atom can have its own feature vector, which is denoted by this X. Okay. Now, the connections of these atoms to each other, which forms the drug molecule, can be encoded in this tensor A. Right. Now, the question is, now you see to the right of this animation, what is happening is I can now act on this graph. What do you mean by acting on this graph? Well, I can then form a deep learning mechanism on the graph by manipulating these representations of the atoms with the weight tensor W. And this is the introduction of the weight tensor W. So if I now act on the graphs directly with the weight tensor, I can update the representation of the graph. And finally, what I can do is then I can predict a lot of interesting characteristics of the graph. Well, what is the toxicity of the drug? What is the solubility of the drug? Can this drug bind to an infectious disease target or not. So all sorts of things I can then handle on the graph directly. Okay, so this is called a graph neural network. All right. So once we know that, the second thing I want to introduce is then how can I generate new drug molecules on this graph? Okay. So then we will consider something called as a generative adversarial network. So you see to the left here, by this generator, which is a fully connected deep learning network, from an injected noise, I can generate a graph. Now this graph corresponds to a drug, okay? This is the task of the generator. Now there is a discriminator which is trained to recognize this generated drug as a viable or a feasible drug or not. Because every random molecule that we generate need not have drug-like properties, right? So then the discriminator is going to say, well, then it will rate it on a very low score. So for example, it doesn't have the properties of a drug. The discriminator will say, yeah, so there's a question.
No, I don't think so. Go ahead.
Okay. So the discriminator, the discriminator is trained on real drug molecules, okay, from real FDA-approved drugs, and that you can get. So the discriminator knows that which molecule is drug-like and which is not. So the generated drug molecules, the discriminator can classify that. So the idea is then to train the generator in such a way that it is going to generate new drug molecules which will actually have drug-like properties. Okay. So if you, so the next step is, once I have a set of these new drug molecules, then I'm again going to pass through a graph convolutional network here, which will output the toxicity and solubility or the binding affinity of these new drug molecules. So the idea is, first, I generate new drug molecules, and the second, I find which score highly in terms of binding affinity or solubility or toxicity and so on and so forth. This can be done from corresponding databases. So once I do that, I train this network on 134,000 stable small molecules, which have carbon, hydrogen, oxygen, nitrogen, and fluorine. So once that is completed, on those two steps are completed, I give a snippet of new drug molecules which may rate highly in terms of low toxicity and high solubility. And again, depending on what features you want it to happen, it is possible that we may generate new drug molecules with those kind of features. So this is just a snippet of what can be achieved. Okay, so here is the halfway mark of my talk, and which is good because I have completed the first two parts, and now I want to come to the main central theme of our talk today, which is infectious disease control at the population scale and within in-house scale. So I'm going to first talk about infectious disease control at the global health, at the population level. So this is an architecture that I introduced after 2 years of working on COVID transmission, right? As soon as it started in 2020, I started working on it. And in 2020, April, I got the first model which had susceptible, infected, recovered, quarantined, and deceased. Remember, in 2020, there was no vaccine. So that was the first, right? So SIRQD. And this, I could really predict the onset and how the virus is going to evolve in 2020. And I got it published in an IEEE Humanitarian Society conference, which was very interesting. It had only SIRQD, okay? Because there were no vaccines. So then what happened is, in 2021, when the vaccines came, my predictions were not correct anymore. So then I introduced this vaccine compartment into this graph, and everything was looking good again. But then again, I got busy with my other network medicine stuff, and then when I run the simulation again, I was again wrong. Why? Because this time the Delta variant came, and you can see that SIRQVD the top part of this model is not ready to take a new variant. So then after a lot of, I went through like 15, 20 models, then I converged on to introducing the third part of this model, the third extension, which is S I1 R1 Q1 I2 R2 Q2 VD model. So the top part will explain the original variant, and the second part was introduced to explain this specifically the Delta variant. Okay, so how this model works, this is of course a dynamical system. So the susceptible is infected with the rate of beta. The infected get recovered with rho. With the rate of tau, they are quarantined. And with the rate of rho, they are also recovered. So the people can recover if they're infected. They can also recover if they're in quarantine. And those who die are infected, go through the quarantine and die with the rate of delta. Now, the vaccination rate is alpha, and the new variant rate of transmission is beta V. So this is how it is. Now, the corresponding differential equations are written to the right? They just mirror exactly what I have explained. I mean, they just mirror that. So I do not think that it is necessary to go through these equations. They exactly mirror what I said. Now, one very big contribution of this graph is this dotted line, okay? Because the dotted line is not straightforward to know that that dotted line exists. So what I discovered here is the recovered people from the prior infected variant could be reinfected with the new variant, right? I mean, there was a debate on that, but I mathematically proved that this has to be the case. That is, the recovered from the previous variant had to be infected by the new variant. If not, the data was not explainable. So this was one of the main contributions of this. The second contribution is, how can I manipulate the number of vaccinated in order to curb the pandemic? Okay, so these 2 points we are going to look. Okay, so how do you find these parameters, right? So the first point is, given the data of infected, recovered, quarantined, deceased, and vaccinated, which was there in Johns Hopkins database, how can I find these optimal parameters? Well, then you form this optimization problem. And here, you take from the 0 is the beginning of the pandemic, right? Somewhere in January 2020. And then you continue till the current time, which was, I think, somewhere in 2021 when my paper was published. So then you want to minimize this cost function, which is the difference between the true infections reported in Johns Hopkins with my predicted infections. Same for the recovered, for the vaccinated, and for the diseased. And this you optimize with respect to the parameters. So I use something called as an Elder-Mead optimization, which is a stochastic optimization technique, which you see here, which forms a simplex over these unknown parameters. And then it manipulates this simplex so that you ultimately reach to the optimized or the optimal values of the parameter, which minimize this cost function. Okay. So once I did that, I found the optimal weights, optimal parameters of this for COVID-19 transmission. And you can now see that the reported infections are given by the thick line, and the fine line is my prediction. Okay, so that is the blue curve. Now the green curve shows the reported recoveries to my predicted recoveries. This purple line shows the actual vaccinations with my predicted ones. And you can see how remarkably they match up, right? And even the deceased, which is hard to see here with the red line. Now, the black line is what the contribution of this model is. It clearly shows you can discriminate the two waves. You see, you could not discriminate these two waves with the Johns Hopkins data, because Johns Hopkins data did not tell you the active infections. It only told you the total infections. So first of all, what you see from this is that I could now clearly discriminate between the two major waves here. So obviously the second one is the Delta caused by the Delta variant.
Abhishek, we have a question here.
Yeah, sure.
Does this assume that people infected with the first variant are equally likely to be infected as with no prior infection? The beta V times the beta T coefficient is the same on both edges.
From
S to I2 and from R1 to R2, I2?
Correct. Yeah. Absolutely. So what I assumed here is that those who were susceptible and were not infected by any of these variants versus those who were infected by the original variant were, equally transmitting, had equal chances, likelihood of getting infected by the Delta variant. Yes, but which could be changed, of course, by changing this edge. But I did not find any reason to do that when I looked at these results here. Okay, so now that I have this one, now look, I am a controls. My main thing is to control, right? So I did not want to predict, right? Because these predictions could be done by many other groups. I think like the White House was always coming up with these predictions, which were done by, I think, Johns Hopkins or University of Washington and Harvard and so on and so forth. So my main thing, yes, I had to predict it. But my main contribution is then to control the pandemic, not to say that what's the trajectory of the pandemic. So how do you control the pandemic? So to control the pandemic, my theory is then that we need to modify the rate of vaccination, because that was the only tool that I had. That time there were no drugs that were efficacious, at least validated to be efficacious in 2021, right? We're talking about 2021. So if vaccine was my only control input, so how do I do that? So what I formed is a energy function of infections. So I1 and I2 are the infections due to the original variant and the Delta variant. So the square of the infections, I defined it as the energy of the infections. So then here is the theory. What it says is that if I can decrease the energy of this function, obviously the energy is I squared. So if I want to go to the bottom, the only way I can go to the bottom is when I is equal to 0, right? So the question is, what is the rate of vaccines which will take me down this infectious sort of bowl-shaped curve? So what is the rate of vaccines that will put me down at the bottom where the infections will go to 0? So how do you do that? Well, you find the gradient of this, right? You just differentiate the I squared term here. And how do you take the derivative? Well, if I get 2i times I dot, you can feed the I dot from the equations here, right? So you put those I dot, substitute it here, and you get this matrix here. And the gradient has to be negative, right? Why do I want the gradient to be negative? Because I want to decrease the number of infections. So obviously I want this gradient to be negative. So in order for this gradient to be negative, I found out what is the vaccination rate that will make this gradient to be negative. And this is the thing that I found out. And then we are going to apply this optimal rate of vaccination into the system and see what happens. Okay. So now you see this black curve is my peak due to the original variant. And the dotted purple line now represents the control vaccination. So you see, the model estimates that there is going to be the second variant coming big time, and see the sharp increase in the vaccination rate here. This is done by the controller that I just derived. So it increases the vaccination rate and estimates that there is a peak that is going to come. It already estimates because it has the full knowledge of the model. And so an increase in the vaccination rate is necessary, is what the model tells. And if it does that, you can see there is no second peak. So that second big peak that we all experienced due to the Delta wave has been completely eradicated because of this increased rate of vaccination. Again, an assumption here is that the vaccine is efficacious to both the variants. I mean, of course, one can change the efficacy by introducing another simple parameter to the system. But this is the controlled vaccination rate that I proposed, and this was published in the Nature Scientific Reports. Okay. So that is the, at the population level, how can one sort of control the viral transmission?
We do have another question if we can take that, Abhishek.
Oh, yeah, sure.
What does the number of new reported infections or the derivative of the smooth number of cumulative infections look like? That's not exactly proportional to the number of active infections, of course, but it seems like it might be a reasonable proxy for relatively coarse-grained purposes like detecting multiple waves.
Okay, so loaded. So what is the number of new reported infections?
Yeah.
Okay, yeah, okay. So I guess what it means, just take this off from here. Okay. So I think the question is that we know the cumulative infections. So that is reported to us. I believe, however, so we are not sure of what is the actual number of infected people at this moment, right? Because this was not reported.
Right.
Yeah, so how close can that be? Yeah, very good question. So, there is, such curves were reported even by the White House. Again, the number of active infections was not there in Johns Hopkins. It was there in some other databases, I believe, but not there here. So, yes, I had to predict this based on the cumulative infections and based on the cumulative vaccinations, based on the cumulative deceased, and based on the number of recoveries. Now, how correct that is now, this is being solved by the optimizer with respect to my model. So if I have to closely match the cumulative number of infections, it boils down to the number of active infections, because without the number of active infections being correct, it will not result in the number of cumulative infections being correct. So by this dualistic model, right, which has the two peaks. But very good question, because, okay, to counter answer your question, what if I had only my previous model, right, which I had, which is SIQRVD, right? No, not the second compartment at all. So then I could not match this. So then there will be only one peak. There will not be the second peak. So if I don't have the second peak, I cannot match the cumulative number of infections. So that is another proof, intuitive proof of that there must be the second peak, and this must be close enough to that. And that brings me to another point here, which I also want to mention, is that you see the green line here. Do you remember that Johns Hopkins stopped giving us the number of recoveries from January 2021? I don't know if anybody remembers, but they stopped. And so this model is helpful then also to keep a tab on the number of recoveries. Okay. All right. So now going back to the problem, now I want to solve this problem inside the host. Okay. Something ridiculous happened at that time, right? So this is 2021, late 2021. So remdesivir was making big waves in all over, right? All of us know that part. All of us know. So remdesivir, to begin with, is not for coronavirus. We all knew that, right? So what was very unreasonable to me at that time was, you take remdesivir, that's fine. It is still an antiviral therapy. You take it and you want to apply it to COVID-19, fine. But you take the exact same dosing, I think it's for Ebola. So you take the exactly same dosing of like 10 doses of 200 milligrams followed by 100 milligrams. I found this makes no sense, because you want to repurpose, that's fine. But you want to blindly repurpose, that's not fine. So I thought, like, we should take the data, if available, systematically, and then find the systematic dosage of remdesivir, at least. So then I thought, how should I do this? So I frantically started looking for some data, any data. At that point, there were a lot of trials that were reported in I think this is also a Nature paper. Yes, I think it's a Nature paper that reported data, real data, okay, of application of remdesivir to acutely affected COVID patients done across 100 hospitals in China. And that was like a gold mine for me. So I immediately jumped on that data. So the data, what the data said is that it gave the data from remdesivir injections to the number of viral particles. However, what was missing in the data was our immune system response. So the T cell response, okay? So I found this model. So what it is showing is this is a virus. The virus is replicating with the rate of mu. It is getting cleared by a rate of alpha. Now the virus is causing an immune response, triggering an immune response with a rate of delta. The immune system in turn is acting on the virus or terminating the virus with a rate of beta. The immune system is cleared with a rate of alpha I, and the immune cells are generated with the rate S. Now this drug injection, which in our case is remdesivir, is inhibiting the viral growth with the rate of theta, and it is getting cleared from the body with the rate of alpha D. So now the question for me was, how can I find the optimal parameters, theta, alpha, mu, beta, right, for this new virus? So again, exactly that the model that I have just said, these equations exactly describe that model. So I don't have to go through these difficulties here. It's exactly the same. So again, I divided the problem into two parts. First, I took this time series data from the published data, based on which I optimized these parameters. That's the first part. Again, I'm not interested in that. I'm interested singularly in controlling the system. So then what is the control variable? Clearly the amount of drug that I'm going to inject. Okay, so once I found the parameters, then I designed a control system, which would give me the exact amount of dosing and scheduling that will not only decrease the virus, but also use the minimum amount of drug. Okay, so let's see how to do this. So the first part, how do I estimate the parameters of the virus? So all I had was the PCR measurements, which was published, and my predicted virus, a number of viral particles. So I needed to minimize this prediction with the actual viral particles that I'll show in the next plot, which was given by this consortium of hospitals. However, my virus prediction is constrained by the dynamic of the system, right? Is constrained by this dynamic that I just showed, how the virus and the immune system and the drug interact. So this optimization problem I solved by this Nelder-Mead simplex method, which I just showed the animation for. And these are the results. So you see the number of viral particles per milliliter. This red curve is the true, the true one is the black. These points are the true measurements. So 1, 2, 3, 4, 5, 6, 7, and 8. So these were measurements taken at the 5th day, I think the 8th, the 12th, the 14th, 17th, 25th, and I think 35th day. These are real measurements taken from acutely infected patients who were treated by remdesivir. And this blue line is the actual treatment by remdesivir. So first 200 milligrams, followed by 100 milligrams, another 10 doses of 100 milligrams, right? So this was the standard of treatment, and which causes these black dots, the virus to decrease. So I fitted my model to this, and the red line shows my model predictions. And the green line shows the number of immune cells per ml. Again, there is no validation to that, okay? Because that data was not available. Now, what are the two clear benefits from this? So the first benefit is that I can somehow predict the onset of COVID, you see? Because when the patients were admitted, they already had COVID. Nobody knew how many days before COVID started. So I could have some understanding of, let's say in this case, 5 days before COVID might have started. The person might have got infected. And this is a rough estimate of how the immune dynamic of our body might have responded to COVID. Okay, so once I did that, the second part was, this clearly felt wrong to me, right? This is for Ebola. So for Ebola, why should we apply the same to COVID? This clearly seemed wrong to me. So then how do I optimize that? So I formulated this optimization problem where I minimize not only the virus, but also the drug. I want to minimize both, right? I want to have a high deceleration of the virus, but I also want to use the minimum possible drugs. So I want to minimize both with respect to what? The same equations, which are the virus, the immune system, and the drug dynamic interaction equations there. So when I do that, how do I optimize? How do I minimize this optimization problem? How do I find the drug? D is the drug infusion and N is the number of days it has to be given. How do I find that? So I use another optimization method, which is called stochastic global optimization method or differential evolution, where you take a population of these parameters and then you keep on improving these populations until there is no further improvement in the cost function. Okay, so this is how you see the animation, how you take the dose and the number of doses, those are the points, and gradually they converge to the optimum where the virus and the drug both are least. So this is the rough overview of that. And when I apply this to the system, you see I got a dramatic result here. What I got is you only need around 100 milligrams of remdesivir, but only 5 times, not 10 times. And by giving these 5 doses, you can actually contribute to the same amount of viral deceleration, very similar to the one before. However, you use much less drugs, and that results in much lower toxicities. Because if you can have this simple relationship of toxicity, then you'll see that the previous dosing, which was 100 milligrams and 200 milligrams, results in a higher toxicity, and the optimized remdesivir administration results in much lower toxicity. And so this was also published in Nature Scientific Reports. And I am pretty sure right now in hospitals, they actually prescribe 5 doses of remdesivir. And my study gives them a mathematical understanding of why it should be 5. So that is the end of today's talk. And again, I encourage you, if anybody wants to visit my homepage, where I have described each of these research and the links to the papers. Okay, thank you very much.
Thanks, Abhishek. Nice talk. We do have one question in the chat here, which is going back to your extended compartmental model. Wanted to know if you've considered having differentiating between the quarantine class for people for exposed and maybe an isolated class for people who are infectious. Have you considered anything like that? Okay, yeah. So another good question. Yeah. So I mean, people had added another compartment here, which is called exposed. So one could add. So then that becomes SEIRQVD. I did consider that in the beginning, but already it was becoming so complicated. Yeah. What happens is, these are stochastic optimization problems. And the more compartments you add, the more parameters you add, then what will happen is there will be multiple combinations of the same parameters that will result in the same, that will justify the same data. That creates additional trouble to us. So then which combination of parameters is better? So my idea is keep it as simple as possible, but also as enriched as possible. So that is the reason I actually removed the exposed from this graph. So let me pick up on that a little bit, because a question that comes to mind that I think does address the tension there, though, is yes, on the one hand, we all want to have simple models where possible, but on the other hand, to the extent that these models get used, if they omit key facts or key aspects of the problem, they could mislead. And so here, for example, it seems clear, right, that for now that we have this possibility of transitioning from the recovered or the vaccinated back to the susceptible. And yes, I would say those are links that there's a double-edged sword. On the one hand, they would definitely complicate the model, particularly because they introduce a form of a cyclic possibility that you don't have. So from a modeling viewpoint, you wouldn't want to have them, but from a policy viewpoint, to explicitly cover that very real possibility seems to be an important thing to talk about. Very important, yeah. And the reviewer, in fact, said the same point to me at that time, that
Yeah, the reviewer, I had multiple revisions before this got published. So reviewer said that you assume that so vaccinated here is, an end pool, meaning there is no further error from here. So obviously, yes, the vaccination wanes, the immunity due to vaccination wanes. So yeah, so again, the time at which when I was doing it, 2021.
Right. Then we all hoped that it would stay this way.
Yes, at that time it was staying. So of course, we now know that after a delay of several months, these vaccinated people will go back to susceptible, but again, their level of susceptibility may be lower than the other group. And yes, so that can create multiple subcompartments to this. And there is no end to such complications, and we can go on. And again, there was the Omicron variant. So how does my model explain it? No. Can it? No.
Yeah.
Meaning it has to be now again extended to IQ. So another I has to be added. And that is for, really graduate students to, I think, take it from there on. Number of variants, this will be that many layers will come here, and you have to keep going on and on. Can you then? A big question for me is: can you predict a new variant coming? Not again, not by this kind of analysis. How do you predict that a new variant will emerge? Is open, and I do not think such model can. That has to be done through genetics. Is what I think.
No, absolutely, and I think that sort of tension is always just a reflection of the question of what is the model for.
Yeah.
If you want to influence policy and get people to think about how to react, then maybe that complexity is needed, even if it does make the modeling harder. And so it's always a trade-off.
Yeah.
We got time for one more quick question. If anybody's got anything, it's just about 1 o'clock. Not really saying much, so I'm just going to say thanks to Abhishek for a nice and interesting talk. And I'll repeat our pitch that if you have anything you want to present to MIDAS, please do get in touch. Otherwise, we'll see you at the next webinar at the end of April.
Thank you very much, Harry. Thanks all.
Take care.